Thanks to the organizers for inviting us, and thanks to Maureen for putting up with a couple of revisions to the presentation. There's that one. Just by overview, Aerpio is a clinical stage biopharmaceutical company developing first-in-class Tie2 activators for ocular indications and also complications of diabetes. Our lead product candidate, AKB-9778, is a potent small molecule VE-PTP inhibitor, which activates Tie2 by basically inhibiting the inhibitor. VE-PTP is the most important inhibitor of Tie2 activation, so you can liken it to removing the brake. You remove the brake, you activate Tie2, and that's activation of Tie2 in absence of Tie2 ligands, angiopoietin-1 or angiopoietin-2, and it also enhances the agonist properties of both ligands. So we think it's the most efficient and effective way to activate Tie2. By activating Tie2, it's well known that you enhance vascular stability. VE-PTP and Tie2 are co-expressed in vascular endothelium, but more recently it's been found that findings from our collaborators and others have discovered that the Tie2 pathway is important in the development and maintenance of Schlemm's canal and is expressed in Schlemm's canal endothelium, and we've recently shown that VE-PTP is also co-expressed with Tie2 in Schlemm's canal endothelium. Our completed phase 2b study in the past year has really bolstered our enthusiasm about Tie2 activation based on two findings. It reproduced the finding in time 2 where we saw a statistically significant reduction in IOP in ocular normotensive individuals with diabetic eye disease. And it also reproduced evidence of improved kidney function in terms of improvement in UACR, and that's with a systemically administered subcutaneous dose. So the pipeline opportunities in ophthalmology are really twofold. One in ocular hypertension and OAG, and towards that end, based on the findings in our phase 2 studies and the emerging science, we have launched into a phase 1b of a topical ocular formulation. I'll go over some interim data in the phase 1b study. And then opportunities in DME based on the time 2 data in patients with DME showing for the first time enhanced effects of combining Tie2 and VEGF inhibition in that patient population. So just a little bit about the science around Tie2 and the conventional outflow tract. The angiopoietin Tie2 pathway plays a key role in the maintenance, both the development and the maintenance of the Schlemm's canal. That's now well known. In the normal conventional outflow pathway, Tie2 is active, and the endothelial cells of the inner wall, as many of you know, adjacent to the trabecular meshwork, where a lot of the pathology in OAG occurs, support the formation of giant vacuoles, which provide a conduit for aqueous humor. By a variety of approaches now, it's been well established that loss of angiopoietin Tie2 pathway activation leads to Schlemm's canal endothelial despecialization, ultimately resulting in generation of Schlemm's canal, increased IOP, and all of the retinal findings associated with glaucomatous pathology. So our hypothesis is if you activate Tie2 via VE-PTP inhibition, you should be able to decrease IOP, increase conventional outflow by targeting Schlemm's canal, decrease IOP, and reduce the progression of glaucoma. These are just some of the studies that support the science. The first finding was a Tie2 knockout, conditional knockout, which surprisingly showed ophthalmos and a phenotype very suggestive of congenital glaucoma, including increased IOP and retinal glaucoma changes. That finding basically started a search among geneticists for families with congenital glaucoma that might be secondary to mutations in the Tie2 pathway, and those have now been found. So the mouse experiment has been shown to be reproduced in humans, and these are heterozygous loss of function mutations in Tie2 that cause congenital glaucoma, and interestingly also in angiopoietin-1, the ligand for Tie2. And more recently, and this is more relevant to the POAG population, is that SNPs in a genome-wide association study have been shown, SNPs and angiopoietin-1, the Tie2 ligand, have been shown to associate with increased IOP in the development of OAG. So these studies really form the basis for our interest, obviously, in glaucoma. One of the questions we had to answer early on is VE-PTP actually expressed in Schlemm's canal? And the answer to that question is yes. If you look at the bottom series of panels there, in the middle panel is purple, and where you see the purple nuclei really indicates expression of VE-PTP. And so we and our collaborators were really the first to determine or show the expression of VE-PTP in Schlemm's canal endothelium. Wasn't a slam dunk because lymphatic vessels don't express VE-PTP, but vascular endothelial cells do. And we also showed that a single topical ocular drop in a mouse eye activates Tie2, and this is shown in the bottom panel as well. Compared, you see phospho-Tie2 in the middle. These are the results from the phase 2 study showing reduced IOP in our phase 2 study compared to the placebo. And in the blue was a once-a-day subcutaneous dose, and in the orange was a twice-a-day subcutaneous dose, so it was a dose-dependent reduction. And that reduction appeared to be maintained over a 12 or 24-hour period at 24 weeks into the study. So these are phase 1B data just released. So this phase 1B study was the first study with a topical ocular formulation of AKB-9778. It was ocular normotensive individuals dosed over a period of 7 days at 3 doses, 5 mg per mil, 15 mg per mil, and 40 mg per mil. And what you'll see here is that at day 1, the placebo-corrected reduction in IOP peaked at about 4 hours, at about 1.5 millimeters of mercury, and then at 7 days, that was actually statistically significant.
Right. Thank you, Kevin. That's great.
Okay.
I'm sorry, we've got to move on to keep on track.